Okay, a few quick notes about, I just got a question about the extra credit quiz. That is not included in your exam grade. That will be in your extra credit column on Blackboard. So for those of you who want every extra point, almost everyone, I think I only saw a small handful of zeros. Almost everyone got between 1 and 5 points. So on that extra quiz. So you can think positively about that. Okay. So exam 2 grades are posted. Do you guys get notification when something's posted, or you just check obsessively? I just always wondered. You can pick up in recitation, pick up the exams in recitation if you want to see them. And if you're not having recitation until next week and you're dying, you can come to today's recitation and go through them. I just don't want to take up time in class. The gene expression topics, everyone's doing really well, coming up with great ideas. So one more group wants to get to tell me their ideas today. Quiz this weekend. Don't forget, I'm noticing a lot of people are starting to forget to take quizzes. That might be like, oh, whatever, but that's going to kill you if you get really sloppy. Of course, you can drop a few. And so if you're at that point of the semester where you're like, I've gotten so many 100% and I want to just stop taking them, I guess that's your prerogative. There's no paper for next week, but there will be an exercise. I'll probably have you guys work through the splicing reaction. And of course, you'll be talking more about the project. So from now pretty much until the end, it'll be focused mostly on your gene expression project. And so between recitation sessions, you guys will be meeting, moving forward. So don't just think this is something you do in recitation, because you're going to be dying. So spend the time outside of recitation to do all the reading and thinking, and then you can use the time in recitation to meet with your groups a little bit more and get my feedback on what your thoughts are. Exam 3 is coming up in about 2 weeks, 4/29. It feels like it's one after the other, but I guess it's better than having all your, I don't know, I think a midterm and a final is fine. I mean, why do I need to do 4 exams? What do you think? Should I get rid of 4 exams and just do 2?
It's kind of nice that you can drop it.
You can drop it, so I guess it's good. I just looked at it and I was like, wow, already? Okay. Here's what exam 2 looked like. It's actually quite a nice average. My first year teaching here, I never was really able to get a 75 average, which is what I think is good. And I'm consistently above 80% in this class. I don't know if that means you guys are brilliant, or my exams are easier, or what. But a handful of people, pretty much the majority of the class, got above a 70. When you add on the possible few extra points from the extra credit quiz, you might be a little bit higher. I looked briefly at the just people's totals, like running averages. There's about 15 people who probably need to be talking to me, or withdrawing, or I don't know. Because if you want to get higher than a C in the class, there's a handful of people that are not looking good. Very small, teeny tiny handful.
The last day to withdraw is today.
The last day to withdrawal is today. Maybe that's my notes here. I don't know. And then there's someone who's like not taking exams but is taking quizzes and is not coming to class. And if you're in here right now, I don't even know who you are actually because I haven't seen you in so long. But anyways, I don't know what they're doing. Okay. But beautiful, beautiful. Okay, so far we've been talking about the basic ideas in gene expression, and then over the last few days we've been going through the transcription cycle and initiation, and that's what we've been doing in recitation this week, drawing out transcription in bacteria and initiation in eukaryotic cells to make sure everyone knows what's going on. The most common mistake I'm seeing is people just like throw all these transcription factors all together, but you have to realize it's a stepwise process. And then there's the idea that transcription is coupled to RNA processing, and that's an important concept to understand, especially as we go through all the processing that happens. We talked briefly about the 5' cap, briefly about splicing, which we'll go into much more detail today, and then polyadenylation if we get, I don't know if I want a dash in there. Polyadenylation if I get to it today. Okay, so these are the different types of processing that happens. Now this is not just mRNA, this is just in general. So you get cleavage, sometimes you have this exonuclease activity that will trim and cleave the RNAs. That's more frequently seen in RNAs that form secondary structure for their function. Splicing, capping, poly-A, and then we'll spend some time talking about editing. And really the whole, when you start thinking about splicing and editing, you can start thinking about how these are mechanisms for generating diversity in proteins. So one gene can result in many different proteins based on how it's spliced and the type of editing that can happen. And so when we talk about gene expression, we're talking about regulation in terms of whether expression is on, off, high, low, whatever. That's one thing. The other thing is you're talking about regulation in terms of when different proteins are generated, or when different mRNAs are generated from one gene that result in different proteins altogether. So just kind of keep this in your mind as we go through this. Any thoughts? Yes?
Is this editing process regular, or like, is it just random? Like
No, this is regulated. Yeah. Is the editing process random? A lot of this stuff you really don't want to be random because then you can have a mess, but this is highly regulated.
So how does it know when it's wrong?
How does it know when it's wrong? Don't forget, the proofreading happens in DNA replication. Not really too, and then there's a level of proofreading with RNA polymerase. Once you have your RNA, you're done. So like if you fold properly, then you'll survive. So if there's some horrible mutation made with editing, it might result in misfolding of the protein, so it might get degraded by the proteasome later on. If there's some editing that causes an error in processing, like something which I don't know happens a bit later about, but let's say it somehow messes with processing, there's quality control mechanisms to make sure improperly processed RNAs don't leave the nucleus. So hopefully there's enough quality control involved where an error in editing doesn't result in an onslaught of crazy proteins. Good? Okay. Any other thoughts? Okay. Okay, so we're talking right now about processing of mRNA. So this is 5' cap splicing and polyadenylation. A really great tricky question for an exam could be something that tests whether or not you understand that processing is different between mRNA and other RNAs. Just saying. Okay. So, addition, so we talked about number one the other day, addition of 5' cap. This is a way for the cell to distinguish mRNA from other RNAs and help with processing, export, and translation. This happens right after the first 25 or so nucleotides are synthesized, right? Because these capping factors are associated with the phosphorylated C-terminal domain of RNA 12-3. That's enough of that. So splicing, removal of introns, right? We learned about this briefly, where we've got this attack of this special adenine on the 5' splice site, and it ends up cutting the RNA. Then there's a free 3' OH that reacts with the start of the next exon, and then this LARIA structure, which contains your intron, is released. This is actually a very useful thing that happens in terms of using it in the lab to look at different things in virology. You can actually, well, even cell biology, molecular biology, you can start to look at in HIV, where you are in splicing of different genes in HIV research depends on whether or not some of these LARIAs are produced. So you can actually look to detect certain LARIAs to decide whether or not some defect in some pathway where it affects the virus. So it's a useful thing to that this cell does for us. Okay, so that seems really simple, but it's not this simple. It's not like just this adenine attacks and that's that. There's a lot of proteins involved, or ribonucleoproteins that are involved in regulation of this process. Makes sense, right? You don't just have random adenines attacking random bases, right? Because there's a lot. How many adenines do you think there are in an RNA? 1? A million? A lot. Kind of depends on the size of your gene. So you have to always ask yourself when we're talking about science, how is this regulated? That should be the first question that comes to your mind. How is this regulated? What's to prevent this random splicing to happen all over the place to generate all sorts of craziness, right? Okay, so this is the lariat structure that's released where you've got your adenine, and then it ends up linked to this 5' end of your intron sequence. That is basically sequence of your intron, this dotted line. And then here's the 3' end of your intron. That's what it looks like at the molecular level. So the splicing reaction is sequence specific. You should already be thinking this, right? There are specific splice sites that are recognized by different ribonucleoproteins. So all introns have a consensus sequence at the 5' splice site. Then there's at the 3' splice site, and then there's a consensus sequence at the branch point. The branch point is where the adenine is, right? So here's exon 1, here's your yellow intron, and here's exon 2. So there's a sequence right here which is partially on the exon, partially on the intron. There's a sequence over here, and then there's your branch point here. And the whole purpose of having consensus sequences is it's a level of specificity for recruitment of something that's going to participate in splicing, okay? If these weren't here, how else would the cell know where to splice, right? Logical if you start to think about it. So splicing is performed by the spliceosome. Spliceosome is made up of small nuclear RNAs, and when they're bound to proteins, they're called a spliceosome. And basically what happens is the point of the RNAs is they're going to base pair with these consensus sequences. And that's how the spliceosome recognizes where the splicing needs to happen. And the whole point is this allows the splice sites, everything to be brought close enough together for the reaction to take place. Okay? How are we doing?
So these sequences are common to all introns? Is that what you said?
Yep. Yep. Because where else would your specificity come from?
Well, I was just wondering if there were different genes and different sequences.
So this is how so the question was, are these sequences common to all introns, or do different genes have different sequences? Well, you have to think about the machinery that's doing the splicing. There's only one, maybe there's some variation, but generally there's splicing machinery that needs to recognize all of these introns that need to be removed. So you're going to have some common way for these spliceosomes to recognize sites. Good question. There are some other this is the major mechanism. There are some other more minor mechanisms that I'm not going to confuse you guys with. Good question. So base pairing between the small nuclear RNAs and the 5', 3' splice sites and branch point mediate recognition of the splice sites by the spliceosome. Don't you like how we always like to own everything? Proteome, transcriptome, proteasome, centrosome. We should have a competition. Who can come up with the most ohms in the shortest amount of time? It'd be like a pie eating contest.
Just
Okay, so now I'm going to go through the mechanism in a detailed way and in a simplified way. This is straight out of the book. So if I'm going through it and you're like, ah, go read the book. Don't ever get bogged down with details. Do not sit here and copy all this because it's in your book. So stop, listen, think, you know. Okay, so you've got the U1 SNRP, which base pairs with the 5' splice site junction, and then you've got your BBP branch point binding protein, which binds the branch point, and U2AF. They recognize this branch point site. That's the adenine that's going to attack your 5' splice site. And then it seems kind of silly, but I guess you need some way to recognize, and it's this BBP that recognizes it. But then U2 comes along and displaces these two, so those two go away, and it's binding to the branch point site. And then there's some other proteins, U4, U6, and U5, this triple SNRP that comes into the reaction. And there's some rearrangements that happen that help to bring things close together. Recognition, change in proteins, bring things close together. Keep that, okay? So it brings things close together because obviously an adenine over here, how's it going to attack there? But if you have an adenine that's brought close, it can attack, okay? So that's the first reaction. Remember, there's two reactions. One is the adenine attacking the 5' splice site, and the other one is this 3' OH that ends up being generated. So so you have these close together, and then you've got other things that happen, which then result in these other. You see how important the details are. I mean, I think if you can understand the things that recognize and understand that there's a lot of SNRPs that come into play that bring things close together so the chemistry can happen, then you're good. These other SNRPs come, and then that helps to bring the 3' OH close to the 3' splice site, and then you've got attacking there, which causes that to be broken and a new bond to be made between your two different exons, and then your lariats release. Okay? So let's just go through it in a less busy way. So you've got the 5' splice site. It's read by two different SNRMPs. There's U1, and remember, SNRMPs are a ribonucleoprotein. So it's a protein that has RNA bound to it, okay? And the purpose of that RNA is it can base pair with these different splice sites. So you've got one RNP, U1, which base pairs here. And then you've got another RNP, U6, that base pairs here. So this helps to increase the accuracy of splice site selection, right? Because you've got this base pairing that needs to happen in order for these splicing proteins to recognize the splice sequence, okay? So then you have recognition of the branch point site by BBP and U2 SNRPs, and this is how you select the correct adenine, okay? So here you have BBP binding, and that actually ends up recruiting U2, and that has these, it also is an RNP, so it has RNA in it, which then base pairs with this consensus sequence surrounding that adenine. Okay, specificity, base pairing of RNA with RNA. Okay, so there's your accuracy of your branch point selection. So after your first phosphoryl transfer reaction, U2, U5, and U6 bring the two exons in close proximity. So the first reaction is the adenine attacking the 5' end, and then so then that breaks that, and then you need the second reaction to occur, which is bringing the two exons together. So you have U6 here, and again, it's an RNP. It's bound to RNA, and the way the RNA is oriented in the RNP allows it to bring, multiple things are happening here, because you're binding to your RNA that's going to be spliced, but then you're also binding to another RNP, which also has RNA, okay? And the purpose is to bring the different splice junctions in close proximity. so the splicing reaction can occur. And then you also have U5 involved. That's another one. It has RNA, and there's more base pairing here. See all that base pairing that happens? It's all just to bring things in close proximity. Okay? There is ATP used, but it's not for the splicing reaction. It's actually for the rearrangement of the proteins involved, the ribonucleotide proteins involved in the reaction. A little detail. Okay, so that's your splicing reaction. Any questions about it? Yes?
When does it become the spliceosome? When U1 and U2 connect?
The spliceosome is really just a way to call all these different ribonucleoproteins that are involved in the splicing reaction.
So it's not like a certain.
It's not like it becomes a spliceosome per se. The spliceosome is just all these proteins together, or ribonucleoproteins together. Yes.
So what happens is through base pairing, steric manipulation occurs so that proximity chemistry can occur.
Yes. Yeah. Yeah. So all that's really happening is base pairing of RNPs with the RNA at specific at the splice sites, so that's your specificity, and at your branch point, are going to cause rearrangements within that RNA structure to bring things close enough for the chemistry to happen. Absolutely. Yeah. So there's actually no true enzymatic activity in the spliceosome itself. Right? There's no cutting. Yes.
So this RNA, is it open all the time, or is something have to open it for it to react with other?
So the RNA is an open. So let's go back a bit. Let's look at a whole picture. So here's your RNA. Okay. So what do you mean by is it
No,
open?
with the spliceosome, you said the space pairing, right? On the spliceosome, is that just open RNA base pairs or nucleotides all the time? Maybe I'm
So the splice. So you're asking about these uRNPs.
yeah,
All
like
these.
U's
Yeah. So all these small nuclear. Okay. So all these small nuclear RNAs that are bound to proteins to form these RNPs. These are the structure is such that they can bind under certain conditions. They can bind to these specific sequences. So why are you asking about opening and closing? Are you
Because
feeling?
is it just exposed nucleotides, just like this, all the
Yeah. Well, I mean, I don't know that the specific structures, the change of the SNRPs. I think that it's just they have a structure that allows availability of the RNA component to base pair with the RNA that's going to be spliced. Or each other, right? Because right here you actually see base pairing between each other. But there's a specific order to the reaction. So like for example, when you go back here, remember these two, you've got to have these two recruited before this can recruit. So there's a level of specificity in the order in which things combine. And also, don't forget, this needs to be in close proximity to the C-terminal domain of the RNA polymerase, right? Because this is happening at the same time.
Because like,
Yeah, I want us to kind of sit on this for a minute. Well, so I think I've got a few thoughts that I want to bring up just in how to think about this. I think it's fairly simple once you wrap your brain around it, but it's the wrapping the brain around that can be confusing sometimes. Okay, so we got through all that. So you have splicing. So just an example of the importance of splice sites and splice site recognition. If you have a single nucleotide change in a splice site, it can result in disease because what will happen is you'll get exon skipping or something. So what can the example here is beta thalassemia and the beta globin genes. So normally you've got these two introns that need to be spliced out. But sometimes you'll get a single nucleotide change that will destroy a normal splice site, so it won't splice there, and instead it'll splice here, and you'll lose an exon. Sometimes you'll get a single nucleotide change that will destroy a splice site and end up activating a cryptic splice site. The cryptic splice site is one that's close in sequence but not perfect. And the way the spliceosome functions is it binds to the best available. So there's this consensus sequence that's really important for the spliceosome recognizing the splice sites. But if there's some error or some change or a protein binding to it or something like that, you might end up with a cryptic, sometimes on purpose, sometimes not. You'll end up with activation of a cryptic splice site, which is just a sequence that's very similar to a splice site, but slightly different. So it's not preferred, but it becomes preferred due to the loss of the preferred, right? So you can end up with that, and then you can also have a single nucleotide change that might create a new splice site and cause a different exon to be incorporated. So this normally was an intron, and suddenly there's a new exon created. So this is why well, mutation in splice sites is a problem. Mutation in splice sites is not a good thing, but sometimes it's used on purpose when we start talking about alternative splicing, which we'll do shortly. Okay, so I just wanted to bring up a few thoughts that we can discuss for a moment. So when we talk about splicing, we think about regulation, right? So if splicing was only regulated by splice site and branch point recognition by these SNRNPs, what prevents recognition of cryptic splice sites and exon skipping? Or, you know, if you think about it, if you have a whole RNA just hanging out there, and what's to say that you're not going to completely skip some splice sites and splice out a whole bunch of exons that you need? And so the way I want you guys to think about this is keep in mind that RNA polymerase is moving along and it's got the C-terminal domain, right? And that's where the splicing proteins are hanging out. And as soon as this RNA comes out and splice sites are recognized, the splicing proteins hop right on, okay? And so there's a level of regulation there. There's a reason why it seems to have been maintained, why having the splice, like processing factors association with the C-terminal domain are so important in the regulation of how processing occurs, okay? So let's say it wasn't like this, and instead you had a full-length RNA, there would be a lot less order to how things could happen. Because remember, splice sites are next to each other, right? And you want to actually recognize the appropriate splice sites and branch points to splice up the correct bits. There are a lot of exons and introns. A lot of introns need to be spliced out of some genes. And if you don't have a way to regulate what's recognized, the wrong things will be spliced out. Okay? Something to think about. Spliceosome assembles co-transcriptionally, but the chemistry actually takes place post-transcriptionally. So all these splicing proteins are hanging out and hopping onto the RNA at the appropriate splice sites and branch points as the RNA is coming out of RNA polymerase. But they're just hanging out there until it's finished, until transcription is finished. And then you have the reaction taking place. Okay? Just an interesting way. I actually hadn't realized that until recently. Any thoughts? Questions? Excitement? Hate? Who hates this class? You're not allowed to. You're not allowed to answer that, are you? Because I have all the power. So if you say yes, actually I love honesty. I love it when people are like, no, you can't. Okay, who likes Friday? Okay, good. I wanted someone to wake up. Questions?
I was going to suggest you should get a better laser.
I know. Well, we can
That's my only
blame Dr. Tsuji. You
I will.
go ahead and blame Dr. Tsuji. Because she suggested, I know, it's like you can see it, but it's just It's
You would have to put it and like hold it. You can't move it.
Yeah, it actually sucks. You're absolutely right. I wonder if there's a way to adjust it. I don't think so. Because sometimes the real laser pointers, you can adjust the size of your point. Anyways, okay. Put that in my evaluation. Needs a better laser pointer. Okay, so any splicing related thoughts? Okay, is everyone like this is so easy? I don't know why she's harping on it. No? Okay, that's good. I think before you learn this, you kind of think it's just there's some enzyme coming along and cleaving some introns out, but that's totally not the case. It's kind of a beautiful dance of ribonuclear proteins. Okay, so I'm going to introduce to you the idea of the exon. It's the exon definition idea, which is it's I guess just a hypothesis with some data, but it has to do with regulation of splicing and identification of the appropriate splice site. So, I've been dying with allergies, oh my word. Most exons are uniform in size. Did anyone ever realize that? I actually didn't either, so it's nice to know. 150 base pairs about. It's the introns that vary widely. So, when you think about a gene, there's a lot of these 150 base pair chunks separated by massive introns. And this is, you've got fly, worm, and human here, and all of the exon lengths are in this 100 to 200 base pair range. Must be important. Okay. Intron size is much more variable. From less than 100 base pairs to 2,000 base pairs, that's the majority, right? Yes?
Would the size of the exons have anything to do with the recognition of the splice sites?
Well, that's where I'm going. Would the size of the exons have anything to do with recognition of the splice site? So that's part of this whole exon definition idea. If you can define your exons in a certain way by being a certain size, by binding to certain proteins, you can start to kind of create these landmarks. So here's the exon definition idea. These SR proteins, and it's serine arginine rich proteins. That's why they're called SR proteins. They bind to exons and mark exon boundaries. So here's your RNA, and here's your cap binding complex at the 5' cap. And these SR proteins are binding to all of your splice sites, and this HNRP also. But and they're involved with marking exons and exon boundaries and recruitment of splicing RNPs. So you can see here's an exon with these SR proteins bound, and here's U2 and U1. And so it helps to identify exons. SR proteins also recognize sequences in exons called splicing enhancers. So they're called splicing enhancers. The other thing that's interesting is that long introns are packaged into HNRP complexes, and this might play a role in masking cryptic splice sites. So that's another level of regulation. If there's all these cryptic splice sites out there, you want to hide them somehow. And the other thing is, I'm guessing having these HNRP complexes, and don't quote me on this because I haven't seen the data supporting it, but it seems logical that if you've got this gigantic intron, having something to condense it might make this splicing reaction go more smoothly also. Thoughts? Heterogeneous nuclear ribonuclear protein, something like that, because I don't know. That's what they say. Okay, so let's talk briefly about alternative splicing. I normally do this later when I talk about editing, but we're going to put this in here now because I got a question about it the other day. which is how is alternative splicing regulated? It's clearly regulated. It's not by accident, right? If that would make any sense. So remember, what's alternative splicing? I haven't been asking any questions today. I've been talking a lot. I'm so sorry. What's alternative splicing? I forgot my list. How about Yaksik? What is alternative splicing? Yes, talk to a friend.
It's like choosing different splice sites to alternate what gene you want expressed.
To choosing different splice sites to alternate what gene you want expressed. Yep, let's expand on that. Anyone want to add to that? Yeah.
It can take a lot of forms. For instance, an exon between vice versa.
So you can skip an exon. That changes your protein. Yep. You can skip multiple exons. So you can have you can create introns, you can have different exons, you can have internal splice sites within an exon. Yes, so this is how we produce many proteins from one gene. Okay, so how is this regulated? So really what it has to do with is recognition of splice sites. If you can recognize a splice site, you'll get splicing. If you can't, you won't, right? Basically. So a repressor, and I haven't really introduced the idea of repressors and activators, which is coming next week. But you can guess what it is, because a repressor is going to repress something to turn something off, and activators can activate something to turn on. So a repressor can block a splice site. So here's an example. This is a negative control, right? Where you've got, here's your primary transcript, and this is going to be spliced out. And then here's your mRNA. It's why you make for it's a very small intron. So the repressor can bind and mask the splice site and prevent splicing of that intron out. And so you have a completely different mRNA, resulting in a completely different protein. You can also have, and so that's more of sterics, just sterics, right? You're not going to be able to recruit your spliceosome to a splice site that's bound by another protein, because it's all about that RNA-based interface, right? So, and then you can have positive control, where you activate splicing of a cryptic splice site. So let's say there's this splice site here, but it's cryptic, so it's normally not recognized. And you can actually bind close to that and recruit the splicing machinery to a specific splice site. Okay? So that's how alternative splicing is regulated in the most basic way. Any questions? What would happen if you didn't regulate alternative splicing? Craziness. It'd be crazy. Right? Then you wouldn't have alternative splicing, because the whole function of alternative splicing is all about preventing or promoting splicing in certain areas. Okay? Okay, should we move on to polydenylation, or do you, yes?
On the other slide, I thought you said that editing had something to do with splice sites also, like changing the base cDNA.
Yep, that was about mutations in splice sites. That was this one. So where if you have a mutation that happens to be, so this is more about mutation and chance rather than purposeful regulation. So if you have a mutation, then you might mask a splice site or create a splice site, and that would result in a protein that shouldn't be made. Yep. So different from alternative splicing, which is purposeful. Okay. Yep.
So could you say that alternative splicing is like a form of fixing a problem where it creates a false, cryptic split? Can you consider that?
So you're saying, could alternative splicing fix
A cryptic.
a cryptic, well, the way to think about a cryptic splice site is it's a splice site that's not perfect, so it's not preferred, and it's only recognized in the absence of the preferred splice site. So when you have a mutation that destroys the perfect splice site, then a cryptic splice site might be recognized. Alternative splicing is all about, think big picture here, okay? You've got something happening to your cell, which results in activation of gene expression. But specifically, a certain alternatively spliced transcript needs to be expressed, right? So you're going to get activation of gene expression, you'll make your RNA. At the same time, you'll probably also be making your repressor protein or your activator protein, which will then be available to bind to and repress certain splicing or activate certain splicing. And that's how like if you want to put it all together to a process, it makes a lot more sense. Yes?
So alternative splicing is more like a subjective selection based off cells actually shape.
Alternative splicing, yes, is a subjective selection based on what the cell needs. It's just like gene expression, but resourceful. Yeah.
If a repressor binds to one perfect splice site, will it go to a perfect site, or will it go to another perfect site that was already there?
It will probably, so if a repressor binds to the perfect splice site, will a cryptic splice site be recognized, or will it go like really far away? That is regulated by the fact that this is all coupled to transcription. So it depends on what is available at the time. So what is coming out of RNA polymerase. There's only so much coming out at the time that's available. Because everything before it's already bound by splicing factors. Everything out it hasn't been transcribed yet. So the next possible, likely cryptic, or who knows, the next possible splice site will then be recognized by the spliceosome. That's a great question. Okay, so this is purposeful. I don't know, I think up until now you may have just learned about splicing as splicing happens. And you get different introns spliced out, and that's it, right? And I guess maybe no one ever, I don't know who's thought about it as why and how, right? Who sits around thinking about this late at night? I know some of you do, yes, I know. We've got some major geeks in this class. And all of you should make sure to go to graduate school. Yes?
We're looking at one gene right now, but I'm trying to fit it all into like the whole cell. There's lots of RNA pol-2s on different genes. So are the same proteins being recruited to the C-terminal domain, like on each of them?
Yep. Okay, so this is good. So let's think about the big picture. When you've got one gene, let's think one gene that's being transcribed at a very high level. You've got lots of RNA pols, all having RNA pol 3, all having a C-terminal domain. Did I say 3? 2. RNA pol 2. All having a C-terminal domain. All having these different, all being phosphorylated at different times by different things to regulate recruitment of these different processing factors. And it's all located close to where the RNA is exiting RNA polymerase, so everything happens at the right time.
But another gene, different chromosome,
Same thing.
same enzyme, but in the same nucleus, are the same like proteins being. So it just seems like you need to regulate exactly what's being recruited to the C-terminal domain.
Yep.
And it just seems like that's a sort of bad way to do it if every gene is going to have the same.
Every gene that's being transcribed at the same time by the same enzyme has the same C-terminal domain, but remember, phosphorylation is what regulates what's going to be recruited when. And then they have their own splice sites. Each gene is going to have its own splice sites, right? So that's very unique. So you might, between adding your 5' cap and adding your 3' poly A tail, have on all these RNA polymerases busy transcribing have splicing factors associated, but they're only going to jump off and bind to splice sites when they're close, when they're available, and it's going to be specific to the gene. Are we good?
I'm getting it.
Okay. Sometimes that was very helpful. I think sometimes it's really helpful to take a step back, because it's easy to get bogged down with you this, you that, bind this, bind that, and you're like, what are we doing? But take a step back and think, okay, cell. cells living in an environment and needs to respond to its environment, which results in gene expression, which results in upregulation of repressors and activators, and all these different kind of things are all happening at the same time. It's kind of amazing that we're alive, isn't it? We should not exist. It's all happening in response to something going on with the cell, up to alternative splicing. Okay? So if you can piece it all together, you're magical. If you get stuck on the details, come see me. Okay? I think you're all magical in your own ways. Everyone's a perfect, unique little snowflake. Okay. Are we good? Should we go on to poly A? Why not? Okay, so polydenylation is signaled by a specific RNA sequence. So it's actually within the DNA is your poly A signal and then gets transcribed to your RNA, and it's in your RNA. And as it's coming out of your RNA polymerase is near that C-terminal domain that has since been phosphorylated and phosphatase, and it has a different modification, so the shape is different, and you've recruited these polydenylation factors, right? So you've got the sequence, and then you've got a cleavage site, and then you have poly A addition. That's pretty much what happens. So how does it happen? Right, so here's RNA polymerase transcribing away, and now there is a different phosphorylation which results in recruitment of RNA processing enzymes CPSF and CSTF, and once you hit the poly A signal that's in the DNA, then these proteins are going to recognize and bind to that 3' processing sequence. Okay? So that poly A sequence is the binding site for the processing proteins. Okay? And so that's why this C-terminal domain is so important. So important. Okay. So then you've got your proteins bound to the processing signal, and then you get other cleavage factors come, and the RNA is cleaved. Okay? So now it's no longer attached to what's coming out of RNA polymerase, and it's hanging out, and you get recruitment of PAP poly A polymerase. So that comes and it binds, and that's actually what ends up generating your poly A. Am I right? Poly A binding protein, then poly A polymerase. Yeah. So then you get poly A binding proteins actually bind along this poly A signal, and you have, wait a minute, something's not right here. Yep. So poly A binding proteins bind along the signal and regulate poly A tail length. And then you get about 200 anucleotides that are added by poly A polymerase. So it's its own polymerase that adds on the poly A tail. And how long it is these poly A binding proteins act kind of like as a molecular ruler. And so then you have your mature 3' end of your RNA molecule. Okay? Simpler than splicing. Okay, so now we have mature RNA, right? You've got your cap binding protein binding to your 5' cap. You've got these SR proteins bound. You've got these HNRP proteins bound. You've got poly A binding proteins, and there's actually a nuclear export receptor. And if all of these things are how they should be, then some things will get kicked off and end up staying in the nucleus, and then you get export of your RNA out of the nucleus, where it then binds to some initiation factors involved with translation, and then will be bound by the ribosome. Okay? But if you're missing cap binding protein, poly A tail, if you have these SR proteins, remember these bound exons, Certain things are not going to allow you out of the nucleus. Okay, so there's a level of quality control here to make sure unprocessed RNAs can't leave the nucleus. So I think we're going to end right here for now. Next week we'll finish this up, but then we're also going to start talking about regulation of gene expression. So for those who like the lactoferron, good stuff. It's my favorite part of the semester. It is, for real. I love it.
Like specific to
Well, so that's what you're going to see. No opera. No, but that's like so I'm guessing 309, 201, med micro.